For those who don't know me, my name is Ludovica, and I'm the outreach coordinator of the Future Vaccine Manufacturing Research Hub, or Vax-Hub. It is my great pleasure to introduce you today to the last webinar of the 2022 Platform Funding webinar series. Before starting, I would just like to remind everyone that this webinar is public and recorded and will later be available on the Vax-Hub website and UCL Biochemical Engineering YouTube channel. So I would kindly request all participants to keep your microphones and cameras off, but please do join the conversation using the chat. This said, I would just start with a brief introduction to this webinar, and then we can move on with the project presentation. So as I'm sure that many of you already know, the Vax-Hub has been supporting an ambitious program of innovative research related to the development, manufacturing, scaling up of vaccines of benefit to low and middle income countries. And we decided to organize this the platform funding webinar series in order to show to the public the great achievements that several feasibility studies funded by the Hub achieved. So these were funded back in 2020 using part of the budget obtained by the Department of Health and Social Care. So today's webinar focuses on mass spectrometry approaches to profile, monitor, and localize host cell protein products in adenovirus-based vaccine manufacture. And we'll present results from a Hub-funded feasibility study that was awarded to Teesside University and supported by UCL Biochemical Engineering. The project aim was to utilize modern mass spectrometry-based approaches on adenovirus products to inform process understanding and improvement, regulatory concerns, and product purity. In this webinar, we will dedicate around 30 to 40 minutes to the presentation of this project, and then allow a 20-minute Q&A session, for which I would like to invite you all to post your questions in the chat. So it is now my great pleasure to introduce our speakers today, starting with Professor Daniel Bracewell, who is Professor of Bioprocess Analysis at UCL Biochemical Engineering, where his research largely focuses on understanding the recovery of biological products. Our second speaker today is Professor James Scrivens, who in 2021 became an emeritus professor at Teesside University that he joined in 2016. And during his time at Teesside University, he established Water Center of Innovation Laboratory and continued his research on biological applications of mass spectrometry, including proteomics, diagnostics, and in particular, shape-selective studies. So a very warm welcome to you both. I would also like to introduce Dr. Salome Desamagalis, who is a Vax-Hub Research Fellow at UCL Biochemical Engineering, and who will be chairing today's Q&A session. And also thank you all participants that join us today. And now over to you, Dan and Jim. Thank you.
I'll let you share the slides, Jim. Maybe while Jim's searching, I'll start with a few words. This project came about, and I'll say a few words about our previous research in the area in a few moments. But it came about originally because Jim and I met. Jim and I both have what's referred to as a researcher in residence award with the catapults. In my case, with the high value manufacturing catapult. In fact, in both cases, with the high value manufacturing catapult. My research in residence award was about thinking about host cell proteins for viral vectors, specifically evolving from work that we'd done on host cell proteins in monoclonal antibody products, and thinking about where we go from there. And it was an event where researchers in residence were able to get together. Jim, are you still having problems? Do you want to send me?
I'm still having problems. It's on the.
I can.
I'm having trouble sharing. Hang on a second.
You were sharing fine, okay, when you shared the.
Come on, here we go.
Looks to be working. Yeah, that's great. Yeah, I don't know if should we go straight to the slides, which I think will slides. So, Jim, as I was saying, we at UCL we'd had considerable interest in the host cell protein area with monoclonal antibody products, going back well over a decade, with some research originally funded by the research councils between myself and Mark Smales at the University of Kent. But I thought I'd just try and summarise in this slide some of the complications and understanding that had arisen from this work on understanding host cell proteins for monoclonal antibodies, as distinct from where we're going to go in this talk, which is looking forward to how it might affect adenovirus and more generally viral vectors. So I think there's a few things I wanted to point out, and these are sort of statements taken out of papers that were occurring during the time we were looking at this. So one sort of very well-known or became very well-known example is the occurrence of a host cell protein referred to as phospholipase B, which was found to co-purify from Chinese hamster ovary cells in the purification of antibody therapeutics. Is found to be a recurring problem. Interestingly, there were some concerns about that, as seen in quote marks here from a paper from Genentech. There were some concerns about its immunogenicity, but even more strangely, I found that some of these lipases could also have problems further through the processes. Not this but other lipases were found to degrade the polysorbate, which is put into the drug substance of monoclonal antibodies to stabilize it, and it was in fact degrading that polysorbate and thereby destabilizing the drug product. So some concerning interactions from what would one might have thought were relatively innocuous low-level impurities. Another example, and an early example from Biogen, and I guess in some ways more obvious, was the presence of protease host cell impurities making their way through and starting to cleave by below less surprising routes the antibody itself and causing degradation. So that I guess is a bit of a prelude, if you like, to me saying there is a lot of complications, a lot of interesting and sometimes concerning details that can emerge when looking down at host cell proteins, individual host cell proteins, rather than what would have traditionally been the case where we're using ELISA to look at host cell proteins as a group and not looking at specific host cell protein issues. So that's one thing I just wanted to flag up and would give you some indication of how we might think the sort of issues we could encounter in the viral vector space. So if we go to the next slide, if you wouldn't. Here was just somehow how we started in our own work to sort of summarize how do we deal with this complexity? How do we say if there are certain host cell proteins that may be of concern? Like in other areas of developing processes, we do some form of risk assessment. But those are gated upon, as we see in the top area here, our ability to measure these things. And their amounts, their identification, and whether they are a concern. So whether whether they have potential to be immunogenic, and there are ways we can predict that once we know the sequence of a protein. So this was, and I don't intend to go through this, it just really summarizes where we got to in our research. It was a paper, a review that we published back in 2015, trying to summarize, as we move from a situation of looking at host cell proteins as a summed totality of impurities to individual host cell proteins, how could we deal with that and not just create a Pandora's box of complexity that wouldn't lead to any improvements in safety and understanding of products. But if we go one slide and into my final slide, that I think will sort of explains the reason for the connection and the reason for starting out to do this work with Jim and his colleagues at Teesside, we can see that we could anticipate, and this is a paper that we published out of the Researcher in Residence activity with Mark Smales and Victoria Smith at CPI and Mike Delahaye at the Catapult, the cell therapy Catapult. We began to think, well, clearly the product is more complicated, and we're in this case showing some examples in the case of adeno-associated virus, but the principle would apply to things like lentivirus and adenovirus, which we're about to see, is that the host cell proteins is indicated in these rather fetchingly coloured pink blobs, could associate within the capsid. They could associate with the capsid structure itself, and different proteins on those capsids. If we look back at the case of host cell proteins and monoclonal antibodies, they are known to be making their way through the process through specific interactions with the product. So they are docking on to the monoclonal antibodies and piggybacking through the process, as many people have described it. That clearly would be more complicated for a viral vector product. It has many more different proteins in its constituents. It may have an envelope. If we're looking at to the left, it's sort of see maybe sort of more of a lentivirus type structure in figure B. And clearly there are even more potential locations and mechanisms by which host cell proteins can make their way through the processes. And indeed, we can start to see this appearing in the literature where people have shown that when they're transfecting and doing different processes using things like PEI, they can find it more difficult to remove host cell proteins, perhaps because they're getting some adherence between those reagents and the product, causing co-purification. Much of this is, so this is based on our, as the title says below, this is lessons learned from the biopharmaceutical area that we think will apply to these gene therapy products. But so this led to us chatting with Jim and his colleagues and starting to understand, okay, well, how do we do this? And there's many issues, and this is a feasibility study that starts out on the issue. But as we can see by these diagrams, some of the issues will be how we identify by which mechanism or how the host cell proteins are associating with the viral vectors. And it's with that, switch over to Jim to take us through where we've got to.
Thank you very much, Dan. Yeah, as Dan said, this came out of a conversation that he and I had when we were researchers in residence. And really the challenge from my point of view is to look at modern measurement methods to see whether or not we have anything to contribute to the understanding of the characterization of these host cell proteins. To start off with, I'll give you a sort of context and background and an indication of the challenges before we show our preliminary results. So the whole idea here is to actually develop measurement methods for us to understand how these vaccine manufacturing takes place and to improve quality of the product that is made. And again, in all of these things, we're doing it with one eye on how we can allow these developments to be used in the low medium income countries to help with the production of these materials. Some context, we're all living in a COVID era, and I just wanted to put the adenovirus in context in the vaccines. And so the two mRNA-based approaches that have been used, the Pfizer and the Moderna, obviously use an mRNA sequence to code for vaccine antigens. And they differ largely in the loading and the time for the injections. Today, we're looking more at the viral vector adenovirus-based vaccines. And the one that we're most familiar with in this country is the AstraZeneca-Oxford approach. There's also the Johnson & Johnson approach that's used quite commonly in the USA. And this is an adenovirus-based approach, and it forms the context of what we're going to talk about this morning. So adenoviruses, for those of you who are not familiar with you, I suspect that's not the audience that we have here, but it may well be the audience that looks at this talk in the future. They're small viruses, they infect humans. They're known that they don't cause disease, but they do cause a mild immune response. And they don't integrate into the genome of the host cell. So the genome includes 4 overlapping genes, which encode both the rep proteins, which are required for the cycle of the virus, and also, importantly, the capsid proteins, which interact to form the capsid, which has a icosahedral geometry. There's also a non-structural protein that's formed during the process. All 3 of the viral proteins are synthesized from one mRNA, which is spliced. That causes us some problems in the proteomics. And our viral vector proteins are built on the VP3 structure with some additional residues added. So our adenovirus proteins, then, we have the rep proteins and the capsid proteins, which have tens of kilodalton molecular weight and form this beautiful icosahedral structure. And the purpose of the work that we have here is not to characterize the virus itself, the adenovirus itself, but to characterize the, if you like, process impurities that come associated with it. So here's a picture of the adenovirus itself. Now, mass spectrometry, which is the technique that I'm going to be focusing on today, can give some indication of the molecular weight, even at the nearly 4 megadalton size of this molecule. But that's not what we're trying to do today. That's a separate structural study. So these adenoviruses are not just used for vaccine vectors. They've also been used in gene therapy and as treatments to cancer. But again, we're focusing today on the use of them in vaccines. So there are a number of advantages. They can infect a broad range of tissues, including those cells that are not dividing at the time. They don't largely integrate into the host chromosome, and you can get stable, high-titer products. There are some disadvantages. The vector may bind to non-target cells. The size of the gene that can be inserted is limited, and the duration can also be limited. When we're looking at the manufacture of these products, we immediately see that there is a challenge in terms of the heterogeneity. So not all of the products are exactly the same. And so we can get a range from a full particle, which is obviously what we're looking for, an empty particle, or a particle that is partially filled. And although this can be seen here with a cryo-EM image, obviously this is not a practical way to measure the process or the quality control as we're going through. The adenovirus vaccine that is being used currently to treat COVID is based on AstraZeneca Oxford product, which itself is based on chimpanzee adenovirus, which was originally isolated from animal feces. The Ad5 adenovirus that we're studying here as a model system, remember this is a feasibility study, and so we're working with those things that we already know quite a bit about, has a similar structure to the ChAdOx. It infects humans and is implicated in common cold. So we're studying the Adenovirus 5 here, not the Oxford product. So just a couple of minutes about the mechanism of operation here. So when we're comparing and contrasting this DNA approach to the mRNA approach, it's not as fragile. The adenovirus has this tough protein coat that helps to protect our delivery material. It attaches to proteins on the cell surface and subsequent to that migrates to the cell nucleus. We have engineered the virus so that it doesn't replicate. The gene released is then copied to the mRNA. The mRNA leaves the nucleus and the sequence is read, and we start to see the spike proteins, which is what obviously the target of the treatment is. So this adenovirus, as I've said, provokes the immune system. This allows the system to react more strongly to the spike proteins that are being released. We also get debris from the vaccinated cells that get taken up by antigen-presenting cells. And these cells themselves then can present fragments of spike proteins on the surface. The helper T cells can then detect these fragments and marshal other immune cells to fight the infection. We also get B cells, which can bump into these coronavirus spikes, and they can then be activated by helper T cells and pour out the antibodies that target the spike protein. And the antigen-presenting cells can also activate other immune cells, the so-called killer T cells, which can then destroy the coronavirus infected cells. So that's the mechanism of the treatment, if you like, but the host cell proteins that we're focusing on here, so where do they come from and what are the things that we need to be concerned about with them? So these are proteins that are produced or encoded by the host organism that we're using. So they're a process impurity, if you like, in a conventional manufacturing sense. But the concern is that they can have an impact on patient safety. And why is that? Well, of course, they can contain a range of protein types, which can include immunogenic proteins. Some of them are themselves biologically active. And the classic method of monitoring these proteins, ELISA, is a powerful method that can detect levels very easily, but tends not to be a protein-specific measurement technique. And so it's less helpful for us in terms of selecting those proteins that can have an impact on patient safety. And so what we need is a method that can provide a more holistic view, a protein-centered view rather than a general view. So the process that we're discussing today in our feasibility study is that developed by Dan and his group in London. And you can see that it's quite a complex analytical system. So with our upstream making the material, we then go through a wide range of purification steps. So having lysed the cells, we're then filtering them, ultra-filtering them, using an actor to capture them, and then a final buffer exchange process. And obviously the optimization of each one of these steps is key to an effective, cost-effective and quality-effective production of material. And so it's quite important to have an analytical or a measurement science approach that can be used at each of these steps in order to monitor any unwanted impurities, such as the host cell proteins. So the challenges in this approach are that the host cell proteins as impurities are very low in abundance to start off with. And as you purify the product, the level of these products gets lower and lower. And so you're looking for a measurement science approach that has enough sensitivity to pick up these very low concentrations of a variety of proteins in the presence of the large amounts of product. And one of the major challenges there is the dynamic range that might be required. So you're looking for something that's got maybe a 10 to the 6 dynamic range, and that's a very difficult thing to develop. So here's a typical blot type product. All I want you to take from this slide is the reduction of impurities as one goes through the different purification approaches. So we can see the viral proteins, the capsid proteins, but we can also see that a significant number of impurities are present, which decrease as we go through the purification process. So what we're looking for then is a technique that will identify and quantify all of the host cell proteins that are being produced during the purification process that we're undertaking. And this is a big challenge. One of the problems with proteomics is the intrinsic dynamic range. So cellular proteins have a 5-order dynamic range variation. Plasma proteins can be up to 12 orders of magnitude, which is beyond any analytical technique. And also, there are quite often abundant proteins present in the same at the same time as very low abundant proteins, and that is a difficult analytical challenge. We can also see, of course, post-translational modifications, which complicate things. Splicing, that I mentioned already, is also a difficulty, but we want to be able to identify and quantify these proteins, and that is a particular challenge. There's also the difficulty of where are the proteins, so the localization, particularly within a cell, the fact that the proteome changes with time, the fact that when one does genomics and we look at mRNA expression levels, there's a very poor correlation between mRNA expression levels measured and the actual concentrations of proteins. And there's very good reasons for that I haven't got time to go into today, but I recommend that you look that up. And of course, the problem that we have with protein levels as opposed to DNA is that there's no PCR process that we can use to increase the concentration. So mass spectrometry has become the established technique for this proteomic characterization approach, and is used extensively to study proteins under very different sets of conditions. And the three main fields are structural proteomics, where we're actually interested in the structure of the protein itself. And obviously, I'm not going to talk about that today, but that's a very important area. Interaction proteomics, and now this is interesting in terms of the context of what we're talking about here, because that looks at how proteins interact with each other and with other systems to produce different functions. But the area that we're going to focus on today is expression proteomics, where we're looking at differential expression, or the levels of proteins that are expressed under particular different conditions. Proteomics experiments are largely speaking based on two different approaches. One is we're looking for particular proteins. So we know what proteins we're looking for, and we're developing methods to do that. And the non-directed, where essentially we're not sure what proteins are being expressed, and we're looking for a method that will enable us to characterize those proteins. And that's obviously the area that we're in today. Again, we can use that just to identify what proteins are being expressed. So that would be a qualitative approach. Or we can use it to get some idea of the relative levels of proteins that are being expressed. So that would be a comparative level. And those are the experiments that we're talking about here. Now you can see under the protein sample there, I have peptides, because the mass spectrometry-based approach is a peptide-based approach. And that sounds a bit counterintuitive to many people. They say, well, why don't you just study the proteins directly? And from an analytical challenge point of view, that is much more difficult than the approach that has been developed, the so-called bottom-up approach, where we take the proteins and we produce peptides. We characterize the peptides, which is a much lower analytical challenge, and then we relate those back to the proteins that are being produced. So this is a peptide-based approach. So we need to extract the protein sample that we're interested in. And again, in this particular challenge, that's one of the problems. It's that the proteins that we're interested in are very low levels. And as we go through the purification stage, they can be very much lower concentrations. We need to produce characteristic peptides, and so we need to reduce the disulfide bonds, protect the disulfide bonds so they don't realkylate, and then digest. Normally we use trypsin, other enzymes have been used to do that. Then having got our peptides, we then need to separate them, and the technique that's been developed for that is a high-performance liquid chromatography of different types. We then separate the peptides, and by using a mass spectrometer, which measures mass to charge, we produce a data set. And the data set consists of the retention time of the peptide through the LC separation system, the mass of the peptide that is being looked at, and then we use a collision cell to break up the peptide into smaller parts. We use a mass spectrometer to measure those masses, and so we end up with something called the product ion spectrum. So the product ion spectrum is the peptide, which has been broken into smaller chunks, and the smaller chunks have been measured. And that is the data set that we're using in mass spectrometry. Now, again, each of those stages is themselves quite complex, and one needs to optimize the extraction and preparation. And the slide that I'm showing here is the summation of quite a bit of work designed to do to optimize the sample extraction and presentation. And so we need firstly, to get rid of the detergents that are present in the manufacturing process as given to us, because they cause many problems in terms of the analytical technique that we're using. We then need to solubilize, or use a detergent to solubilize the system. We need to reduce the disulfide bonds, protect the disulfide bonds, and then digest with an enzyme. And we use trypsin, which cleaves after arginine, after lysine, unless it's next to proline. So there's a well understood mechanism of operation there. And you can see that I'm talking here about two main sample types that we've looked at. One is after the TFF purification step, and one is after the TFF and the AEX step, as shown on the purification process from Dan's group. Now, having collected that data, we then need to decide what to do with it. And this slide summarizes all of mass spectrometry-based proteomics. And I'll just talk you through some of the simple approaches. So having got our protein mixture on the left-hand side, we then do our digestion to the peptide level. The peptides are separated using liquid chromatography. We then have the mass spectrometry stage of analysis, where we collect the molecular weight and the molecular weight of the fragments, the product ion spectra. And then we collect that database, and we can then use that to search against the database. So what database do we need to use? Well, we obviously need to choose an appropriate database, searching all of the proteins that have ever been identified in the world. Has been done, but it's not, generally speaking, the most effective way of doing it. So we need a genome of the system that we're looking to study. And then we can use a number of different bioinformatic approaches to predict the proteome that might be produced by that genome. We can then, from the proteome, do a simulated in silico digestion to predict the peptides that might be produced. And indeed, we can then predict the product ion spectra that each one of those peptides might produce. And so we have an experimental database and a theoretical database. And by using software algorithms that have been developed, we can then relate the two together and identify the proteins that have been produced. I told you that we wanted to do more than that. We wanted to actually quantify them as well. And then we can do that either by isotopically labeling them or by using an approach that I'll discuss in a moment, which is much simpler. So we're using a data-independent mass spectrometry approach, where we're not choosing the peptides that might be produced. We are using high-resolution mass spectrometry, and so we are mass measuring the intact mass of the peptide to better than 5 parts per million, and also the product ions to better than 5 parts per million. Why is that important? Because it reduces the complexity of the search and enables us to get a more targeted outcome. So in our bioinformatics search, I've already told you we need to choose what database we're using, what software approaches, what search criteria, how many peptides do we have to identify before we can be sure that we've identified a particular protein. We need to work out how we're going to quantify that, and then what pathway analysis we're going to utilize. So in this case, the protein databases that we chose, an AD5 database, human embryonic kidney database, and the so-called CRAP database, which is based around the protein impurities that one gets by touching the sample, and they're always present, and so we need to get rid of those. Our search software, we're using a company called Progenesis to do our searches. The criteria that we're using are automatically selected based on the quality of the data that we're using. And our quantitation is the so-called high 3. So high 3 uses the fact that the concentration of the protein is proportional to the sound intensity of the most intense peptides. You can use any number you like. Traditionally, or most commonly, we use the top 3 peptides. So the sound intensity of the top 3 peptides of each protein is proportional to the concentration of the protein itself. This is described in the paper by Silva et al. In 2006. This is good because we don't need to put a calibrant inside, and we don't need to label the proteins. And it gives a quantitation of all the proteins that are present. For those of you who want to know more about mass spectrometry-based proteomics, particularly this bottom-up proteomics, I recommend this particular recent paper that talks about the advantages and disadvantages. So finally, let's go on to some results. And so I told you that we looked at 2 particular batches, one after tangential flow filtration, and one after tangential flow filtration and the anionic exchange chromatography step. The instrumentation that we're using is Waters-based. It's a QTOF, quadrupole time of flight combined mass spectrometer with high performance liquid chromatography. So these instruments are widely available in many industrial and university establishments. So now let's look at some results. So in this batch A, you can see I've shown 2 results there which show the difference when we vary the concentration of the enzyme that's digesting the protein to the overall protein concentration. This is something that can significantly affect the quality of the results and needs to be optimized. So if we look at the results on the left, we have 7 AD5 proteins that have been identified, which is actually what we expect. We have 293 human proteins. Now, first thing I want to point out is that is quite a large number, and there's quite a wide variety of types there. And so one can understand why it's essential to understand exactly what proteins are being produced so that we can look at their immunogenicity properties and other areas. And you can see that we've identified 7 proteins that are essentially impurities in the process. If we compare those results to the higher level of, the lower level of trypsin to protein, or that one can see that the results are similar, but not as good as the 1 to 50 results. As we go through the process and we end up with the increased purification, so this is now the second batch, you can see that the numbers of proteins that we're identifying drops off. And this is why we need to optimize the process. So this is using the same conditions that we saw before. We're now needing to optimize this process in order to be able to see more of the human proteins. You can see here that we're really only identifying 1 human protein here. 3 are part of the concentrations. And so this is why we need to do the research, do the optimization of the various stages in order to improve the process. And so here is our best analysis. So we can see here now we're at the batch 2, and we have the 2 different levels of purification. And you can see in the AD5 area, we're essentially identifying all of the capsid proteins. And you can see that in the human proteins, you can see that we're identifying between 60 and 70 human proteins with a relatively small number of impurity proteins being present. And so we have here now a viable, reproducible method of identifying these proteins. So the viral proteins that are identified are exactly the ones that we'd expect. And you can see that the unique peptides for each of them is very high. So this is what you get if you have a high concentration of proteins. Obviously, with our human proteins, the criteria of these is significantly lower. So we now have a method of identifying and relatively quantifying the proteins that are produced during the process, even after the purification process. So let's compare that with the existing methods. So if you're developing a new method, you have to have some advantage over the existing method. And the ELISA method has been very commonly used, and that's very sensitive and reproducible, but it's not individually protein specific. So it's unable to identify individual proteins. You can see that we have 60 or 70 or more human proteins that are present, and obviously only some of those will give rise to concern over their immunogenicity properties. So our mass spectrometry-based approach, our bottom-up mass spec approach, can identify the individual proteins. We can get the relative concentrations, and we can study them even when we have the purification processes in place. So in summary then, the vaccine samples manufactured in London that we've studied, we have obviously all of the conventional 85 proteins identified, but also a significant number of HCP proteins. Those protein concentrations drop through the purification process, and so we need a very sensitive high dynamic range technique that will enable us to characterize them and understand them. And when we go through the HCP proteins that we've identified so far, the things that jump out at you are that they include a significant number of heat shock proteins, chaperone proteins, and cytoskeletal proteins as obvious protein classes. But obviously, in terms of what Dan talked about at the start, about the importance of the regulatory and health concerns, you need to study each one of these to see whether or not they would have any health problems. So finally then, we've been able to characterize these processes. Obviously, there's a lot of work still to be done. So now, having developed this method, we can aid Dan and others with their process conditions. So we can tell them whether or not their purification stages are cutting down on these proteins and which ones are getting through. We now, having developed the method, can study where these proteins are. Are they inside the capsid? Are they outside the capsid? How are they being presented? And now we can also study different types of proteomics experiments where we can select any proteins that have been identified as potentially causing problems and just quantify those. So we don't have to now identify all of the capsid proteins, all of the HCP proteins. We can focus in on any proteins that have been selected as causing any potential problems and just measure those as we go through the process. And so I hope I can convince you that we have a robust, good method that can be used to do that, that has significant potential to being used on the manufacture of these types of systems in the future. Thank you for your attention.
Thank you so much, Professor Jim and Dan, for the lovely presentation. The Q&A session is now open, so please put your questions on the chat or raise your hand if you have a question to ask. Okay, I have already one from Adil. I will read it for you. When starting out populating a HCP database for a given host, if needed, how do you accommodate for effects of heterologous protein expression that may, after post-translation modifications of endogenous proteins or abundance, that is to say, due to metabolic stresses and folded protein responses? Also, to what extent are you concerned about species that are undetected in mass spec? Is there any way to get a feel on the size of the problem?
Yeah, okay, so the first thing you can do is obviously you have your peptides that come out. You know how many of those you have identified. You know then that you've used those to search against the database. In any system, there will be a number of peptides that have not been identified. So they have not been identified as being coming from the system that you're studying. Now that can be for a number of different reasons. Most commonly, particularly in human proteomics, it's because of post-translational modification. And so one can use bioinformatics to ask the system to look for post-translational modifications. So glycosylation, phosphorylation are the most common. One can also, in important systems, one thinks of the manufacture of recombinant proteins, where the identification of every peptide is essential because of the regulatory. One can look at those peptides that have not been identified and interpret them either by hand, essentially by human interaction, or by using software that has been developed. PEAKS is something that we use a lot, that will identify the peptide even though it has not been identified in the system. And so you can look, as well as identifying the systems, the problems that we have, you can interrogate your data set to look for those things that have not been identified. And then you can either use other protein databases to see if they come from somewhere else, because you can always add on another protein database, or you can interpret them from scratch and see whether there is a result of post-translational modification.
Thank you, Jim, for your answer. Adil, I think the answer was complete. Okay. Please, Steve, go ahead.
Yeah, thanks for that, Jim. That's really nice. I just had a query on the quantitative aspect of these things. So in the example you gave, the sort of, you were detecting about 70 proteins, both off the TFF and the ion exchange, which sort of almost implies that the ion exchange doesn't really do very much in terms of.
No, that's
so
not
is there a quantitative aspect in this?
I mean, essentially what we would look at, there's 2 points to make there. So first of all, there is the optimization of the experiment was different for the 2 stages. So we did not, under those, if you remember, I showed you one slide that showed how if we used our existing method and we went through the 2nd stage of purification, we only ended up identifying 1 protein. And so we had to improve the experiment to see those numbers of proteins. I also told you that the experiments we were doing there were relative quantitation. So what we were doing was looking at, I could tell you in those 60 proteins what the relative concentrations of each ones were. Now, a separate experiment, which we didn't do, but we do normally, is you can add a protein that is not present in the system at a known level, and you can get a more absolute quantitation approach. So it is possible to do what you're saying there. We did not do that in this experiment here, but we do it in many other cases. So if you essentially dope the proteins that you've extracted with a known amount of a protein that doesn't exist in the system, and you use these sum of peptide concentrations to quantitate, then within about 5 or 10%, you can get a absolute level of protein concentration. And if you did that on the different stages of separation, we can see that there has been a very significant purification of the HCP proteins going through that stage.
Okay, thanks very much for that clarification, Jim. That's important. Thank you.
Any other questions from the audience? Okay, while we're waiting, I have a question. It's a general question regarding the application of MS. So even though there has been an increase on the application, clinical application of MS, our challenge, like lack of standardized methods or even lack of automation being tackled?
I think that very largely now, I think, has been addressed in the sense that if you look at manufacturing, obviously the pharmaceutical industry has used mass spectrometry for many years.
Mm-hmm.
The biologics aspect now of the pharmaceutical industry, which is now obviously the most important, mass spectrometry has become a very important component of the manufacturing process. And the last 5 or 10 years has seen the introduction of a number of what I'd call process-specific mass spectrometers. So mass spectrometers that have been designed and sold essentially with one purpose in mind, which is to help with the manufacturing process. And so these dedicated instruments that are now used extensively have built-in automation. So this is still being done on a sample-by-sample basis, although a number of people I know are working to see whether or not a more continuous experimental approach might be used. But the introduction of relatively low cost, I mean, relatively being, £100,000 or so instruments, which are dedicated to this, to biologics manufacture essentially, is becoming now much more common in the pharmaceutical industry.
Mm-hmm. Thank you. Just follow up on what you mentioned about costs, because you said that at the beginning that it would be to be applied on LMICs. So how do you envision to make it more cost effective? It's more trying to turn the MS even more easy to combine with other techniques, or as a system that could give us more information rather than mass, for example, shape as well, or other parameters?
I'm passionately interested in shape, but that is a much more challenging and expensive mass spectrometry experiment. In terms of what you just asked, I think there are two major things. Firstly, we can use the mass spectrometry approach that's being developed here in order to influence and advise our colleagues in the LMIC countries as to how best to introduce the purification stages. So we can study the purification stages ourselves and give them the information to introduce there. But secondly, I think that a mass spectrometry center or a mass spectrometry instrument in one of these countries, using the techniques that we have now developed and optimized, and can use it to study and to characterize the quality of their product, which doesn't need to be done on a day-by-day basis, but on a batch one-off basis. And I think mass spectrometry is now becoming so widely introduced, even in LMIC countries, that the ability to be able to use an already developed method to address the product that they're making should be possible.
Thank you, Jimmy. We have another question from Adil. Bit off topic, but you mentioned at the start being able to run 3.9 megadaltons assembled particle. Interested to hear a bit more. Is this native MS?
Yes, this is native MS. I would address you to the work of Albert Heck and others, but Albert Heck is a good place to start, who have developed mass spectrometry approaches that enable you to study the intact capsid 3.9 megadaltons, and in fact even higher than that in a mass spectrometer. So the electrospray process, which takes the sample from solutions. I know a lot of people are worried about the fact that mass spectrometry operates in the gas phase, but there is a solution phase memory in the electrospray process. So the electrospray process, we take the solvent off the system. It goes up into the gas phase where it's a protonated species. Is the solution phase structure is maintained for a number of 100 milliseconds, and so enables us with the rapid mass spectrometry analysis to be able to look at the intact mass. Now the challenge is that one can look at the intact mass and get an idea that it's 3.9, but there's actually a range of molecular weights that are produced. And most questions that people want to know is, what is the load? You know, what is inside the capsid protein? And there, of course, you're looking at differences in mass that are quite small. So it's one thing being able to look at the intact capsid in the gas phase. It's one thing being able to try and decide whether it's empty or full. Now, a number of quite exciting experiments have been done, and I would address the work of Albert Heck and others if you're interested in that particular area.
Thank you, Jimmy. Is there any other question? If not, I'd like to thank again to both professors for the amazing presentation, and back to you, Ludo, for the final remarks.
Great. Thank you both, Jim and Dan. That was a great talk. I would just like to conclude by sharing my screen again briefly, because this is the end of our platform funding webinar series. So I would like to thank all presenters for giving really great presentations on their feasibility studies, and also all participants for actively contributing to this webinar series. I really hope you enjoyed it, and just to let you know, we will soon publish all webinars on the Vax-Hub website and UCL Biochemical Engineering YouTube channel. So if you will ever want to watch any of these webinars again, you can find it there. And this said, again, thank you all very much, and I hope you will enjoy the rest of your day. Thank you.